Okay, so we'll begin now. Hello, everyone. I hope you're all doing great. I hope CVP went well and Meal is going well. We have MOL. I know MOL is a bit tough, but it's okay. We can get through it, and hopefully these past sessions can be easy for you guys. Okay, so for today's past session, my name is Ghena Sharia. I'm a third-year student. And guys, feel free to ask me any questions after the session if you're watching this as a recording or during for sure. And yeah, here's my number and here's my info. Just reach out, ask me any question you want, whether regarding this lecture or afterwards. I genuinely really don't mind. You can reach out with anything and I'll be more than happy to answer. So for today's lecture, I'm going to be starting and talking about the respiratory chain and oxidative phosphorylation in MOL. This lecture is a part of a series of lectures. So in general, the objectives are very simple. We're going to be talking about the components of the electron transport chain. Then we're going to cover a very important concept that comes with the electron transport chain, known as the oxidative phosphorylation. Then we're going to look at the inhibitors of the electron transport chain and how they work exactly, and then the uncouplers of the electron transport chain. And then we're going to differentiate between both because they're very different, they're not the same. Before I begin, I'd just like to say, the way I'm going to do this is that we have so many diagrams in this presentation. I'm going to be explaining over the diagrams, and then I've included my explanations later on in the slides, so hopefully that helps you guys. So I just mentioned that this lecture is a part of a series of lectures, and there's multiple stages that come when we're talking about cellular respiration. So first of all, we have the glycolysis process, which is when you break down the glucose to produce pyruvate, and some ATP. Usually it's just 2, and we call this process glycolysis. And so this is known as the first process that happens in cellular respiration, which I believe you've already covered by now. And then number 2 is when we produce acetyl-CoA from the pyruvate molecule, and that's usually the second stage. And then we go through the citric acid cycle, or also known as the TCA cycle. And then eventually we reach to our point today, which is what we're going to be talking about, the oxidative phosphorylation. Now, before we go ahead and discuss the oxidative phosphorylation and everything, I want us to try visualize where we are in the cell, okay? So where are we now? Yeah. Okay, so where are we now? We're obviously in the cell. So we're in the cell, and then in the cell, we're in the mitochondria. But where are we exactly in the mitochondria? And this is what I'm trying to reach to. So basically, we're in between the space, which is known as the intermembrane space. And that comes in between the inner mitochondrial membrane, which is this membrane over here, this here, and the outer mitochondrial membrane, which is this area. This area in between, we call it the intermembrane space. And this is our focus point for today's lecture. This is where the electron transport chain is going to be happening. As you guys can see, all these channels in the inner mitochondrial membrane, and they're going to be pumping protons into this space, and then it's going to go back into the matrix and so on. So in terms of location, we're in the area between the inner mitochondrial membrane and the area, between the inner and the outer mitochondrial membrane, also known as the intermembrane space. So now that we've understood where we are in terms of location, what the general layout of this whole process is, we're going to actually begin discussing some important concepts, and then eventually we're going to gather all of that into one diagram and understand the process as a whole in terms of a story. Okay, just to make things easier and simpler for you guys. Before we go ahead, we need to always understand that in the oxidative phosphorylation process, we have important reactions taking place. They're known as the redox reactions or oxidation reduction reactions. Now we know that the redox reactions, they have to do with the transfer of electrons. So when electrons are transferred, for example, a species loses electrons, these electrons don't just disappear and they stop, they just go away. They're always gained by another species. So any electron lost by one species is gained by another species. And the process of losing an electron is known as oxidation. The process of gaining an electron is known as reduction. And I brought you guys an example here that we're going to see later on when we talk about the electron transport chain, which is when we have hydrogen atoms. And then we have the electron carrier, NAD+. Okay, this over here. NAD+ has a positive charge, and so it needs an electron to balance itself. And the hydrogen ion has two electrons because it's H2. So what happens over here is that we have NAD+. Oh, sorry, guys. This is actually supposed to go here. Okay, so we have the NAD+, which needs an electron, and then we have the H2, which is going to lose an electron. So what happens here is that the NAD+ gains an electron by gaining a hydrogen atom, and then the H+ loses an electron by losing a hydrogen atom. So now I have, before I had two hydrogens, and I have an NAD with a positive charge. Now I've neutralized everything. The NAD has gained an electron, which I call what? Reduction, because it gained an electron. And then the hydrogen ion lost the electron, and that I know as the proton, the hydrogen atom, okay? Now, an easy way for you to remember it, I gave you guys this very quick mnemonic. It's called oil rig. Oxidation is loss, reduction is gain. Okay, and I've colored it green so that in your head, خلاص, you can always remember that reduction is gain of electrons, oxidation is loss of electrons. Does that make sense? So redox reactions, they're basically transfer of electrons. When an electron is lost from one species, it's always gained by another one. It never just disappears. An example on that is hydrogen atoms. When they give off electrons to the electron carrier, NAD+. Sorry, just a second. I don't know why it stopped. Okay. Did my screen appear again or no? This is weird. I'll try again. Okay, so now we're back. So as I was saying, these are the redox reactions, and I gave you guys a very easy mnemonic to remember that when they ask you which species which species underwent oxidation, it's always going to be the species that lost the electron, which species underwent reduction, and that's always going to be the species that gained the electrons. And how do I remember this? Oil rig, remember, okay? So in our example, which one underwent oxidation? It was the one that lost the electron, and hence it's the hydrogen atom. It went oxidation. And then the one that gained an electron is the NADH, and that's the one that underwent reduction. So now we've mastered this concept. Let's go ahead and move on. I've put here some questions for you guys when you're reviewing this lecture. It's like a quick recall type of questions. It's just for you to remember exactly the concepts as they are, and they're not there to test your knowledge. It's just for you guys to make sure that you're memorizing properly. Okay, now we've mastered this concept. Let's move ahead to the electron transport chain. Now we're going to discuss the electron transport chain as if it's literally different transporting vehicles, and this will make it more easy for you guys to visualize things and understand them. Because it's basically all just like a connection between different things. So what is the electron transport chain? It's also known as the respiratory chain because it's a very important part of respiration. It's the final step. Now, where is it located? It's found in the inner mitochondrial membrane, as I mentioned previously. And in terms of function, a very summarized function of it is that it's called an electron transport chain. So it accepts electrons, transports them from one carrier to the other, eventually reaches a final carrier where it can actually produce ATP from the movement of these electrons with a few other additional ideas. But this is the main idea that you guys need to know, is that the whole point of this whole lecture and the electron transport chain is for us to accept electrons, transport them through the chain, and this will help us create ATP, and I'll tell you exactly how throughout. And a very important thing to remember is that we have multiple electron carriers, and they basically alternate between reduced and oxidized states. Based on what do we know if they're reduced or oxidized? It's whether they gained an electron, they'd be reduced, or they lost an electron, they'd be oxidized, and so on. And in the end, the electrons are finally passed at the end of the chain to oxygen, and this forms water. Now, these ideas may be like a bit distracting for you, and it's like you really don't get things, but just wait for me now. I'm going to be explaining things on the diagram, and it's going to make sense, inshallah. So these are just some more questions for you guys, just after every big slide, you guys can just like remember things and make sure that you memorize them properly. So now let's come to our first component of the electron transport chain. I call them the taxis, okay? Now, why are they taxis? Because they're basically transporting things and they're bringing them and they're dropping them off. Now, electron carriers, from their name, they carry the electrons just like taxis carry passengers. They carry electrons and then they drive them around and then eventually they drop them off at the first complex in the electron transport chain, okay? So always remember, the electron carriers are exactly like taxis. And what are two very important examples we have in the respiratory chain? We have NAD+, which is the first, NAD+, which is the first electron transporter, and then we have FAD. Okay, so these are our two taxis for today. Now, NAD+, in general, electron carriers in terms of their, like, let's say, chemical structure, they are organic cofactors or coenzymes, and they help enzymes that are needed for catalytic activity. They help activate them and ensure that they're working properly. And in our case, and in this context, we care about them in terms of electron acceptors and carriers. They just carry the electrons, they drop them off. So NAD+ is derived from the vitamin niacin, B3, and FAD is derived from the vitamin riboflavin, which is B2. Just memorize this, just in case they bring you a very fast recall question. This would be like an easy mark for you, because khalas, you've memorized. NAD+ comes from niacin, which is vitamin B3. FAD comes from riboflavin, which is vitamin B2, okay? And since they accept electrons and then they lose them, what do they undergo? Oxidation reduction, okay? Now, I explained to you guys why I think of them as taxis, because they deliver all the electrons and then they drop them off at the electron transport chain so they can move through it and eventually produce ATP. Okay, so what are the electron carriers? There are taxis for today. They're going to carry the electrons, they're going to drop them off, and so on. Now we come to our very important diagram. But before I come to this, I just want to quickly just like go over each complex that we have. In this diagram, we're talking about this, as you see here, guys. It's the electron transport chain, okay? So we have complex 1, we have a coenzyme Q, we have complex 3, and then we have cytochrome C, and then we have complex 4, okay? These are the components of this chain. And then finally here, I have ATP synthase, which is also a very important part of this. Now let's talk about them before, and then we can come back to this diagram and actually discuss them in terms of how the process happens. Now, the ETC components, I told you guys, we're going to be talking about things as if they're like transporting vehicles, let's say. Now, complex 1, which we just saw, also known as NADH dehydrogenase, is our entry gate. You know the taxis? They're going to come, they're going to drop off the electrons at the complex 1, the entry gate. So the electrons are going to go through the gate, they're going to enter, and that's the first stop they're going to reach. And then eventually they're going to start going from complex 1, and then they're going to go to coenzyme Q, which is also known as ubiquinone. Now, guys, know the names, coenzyme Q or ubiquinone, because one question can be asking, and it can say coenzyme Q, and then or it can even say ubiquinone. So they can ask you like about the same thing, but in two different names. So know both of them, ubiquinone and coenzyme Q. Now, coenzyme Q is like our shuttle bus. It's not a complex, it's just there in the middle. It is able to move around the bilayer, but at the same time, it just carries the electrons and makes sure they go to complex 3. So complex 1 is our gate. The electrons were dropped off by the taxi. Khalas, they've reached their destination. They go through the gate. And when they go through the gate, they're eventually going to go to coenzyme Q, which is our shuttle bus. This shuttle bus is just going to be moving around, and it's going to be moving around and carrying these electrons until they reach complex 3. In complex 3, they're then going to go to the other component, which is cytochrome C. Cytochrome C is our second shuttle bus, okay? Which will transport the electrons to complex 4. Complex 4 is also known as cytochrome oxidase, and that's our exit gate. This will be our final electron transporter. And this eventually will let the electrons leave, and the terminal electron acceptor in this case will be what? It will be oxygen. And when oxygen accepts an electron, it's going to be in its unstable form, and hence it's going to join with the other hydrogen ions to form water. Okay? Now, I've went over things very fast. Let's go back to the diagram and understand things in a more visual way. And why did I call, now before I go, why did I call them shuttle buses? Because they are mobile, meaning they don't have a specific place in the lipid bilayer. They can actually move around. And once you can find them in this specific area, and another time you can find them that they moved along the lipid bilayer. That's why I called them shuttle bus. So you guys can remember that they're mobile carriers. They're not in one specific place, rather they move around. So we've discussed these 5 components. Let's go back and try and understand things according to this complex one, okay? So now we're at complex one. طيب. The taxi came. What are the taxis? They're NAD and FAD. NADH is going to give off an electron, and FAD, كمان, if it were there, would give off an electron. So in the case of NADH, it's going to give off an electron. When NADH loses an electron, what is this called? Oxidation. And then who's going to gain the electron? It's complex 1. So complex 1 is going to get reduced, NADH is going to get oxidized. And now you will find that the electron is actually found over here. Sorry, okay. Now the electron has been dropped off by the taxi, NADH, and it's in the first complex, known as complex 1, okay? And then it's going to move from complex 1, and it's going to go to coenzyme, what? Coenzyme Q. We said coenzyme Q, as you can see here, it's this small structure, and we said we called it what? We called it the shuttle bus because it's mobile, it's a mobile carrier, and it simply just allows the electrons to pass, okay? And it ensures they reach complex 3. In complex 3, they're going to go eventually to cytochrome C, and then in cytochrome C, another shuttle bus, so it's a mobile carrier as well. Then it's going to go to complex 4. Complex 4 is what? It's our exit gate. So this is the entry, and then this is our exit. Why do we call it the exit? Because this is where the electrons leave the chain. And how do they leave the chain? They by the presence of our terminal electron acceptor, which is the oxygen molecule. So the oxygen is going to gain these electrons, and when it gains these electrons found over here, it's going to combine itself with hydrogen atoms to produce water. Okay? Does this make sense? Perfect. Now you guys can realize that we've actually missed one part, and that is the ATP synthase. Now ATP synthase, I want to first talk about something for you guys to understand how ATP synthase ends up producing the ATP in the end. Because till now, all we've talked about is how the electrons move from one complex to the shuttle bus, and then complex, and then shuttle bus, and then complex, and then they leave the chain. Now, what does that have to do with the ATP, and how is it actually produced? Now, there's exactly a story, okay? This story, let's say, it's a story behind how ATP is produced from this chain. Like, okay, yes, the electrons have moved from one complex to the another, but how did this exactly give us the energy that we need? Now the story is titled Chemiosmosis, okay? Chemiosmosis is known as an energy coupling story. What I mean by that is you guys can realize that the electrons have been moving from one complex to the other, and the NADH gave off an electron. However, the NADH, when it actually goes through this process, the reaction looks exactly like this. Okay. So this is what the reaction looks like. So when the NADH, which is our taxi, comes and drops off the electron, it doesn't directly just give off the electron. It also releases the hydrogen ion. So when the NADH loses its hydrogen, the hydrogen breaks into a proton and an electron. Where does the electron go? The electron goes through complex 1 and then moves from one complex to the other, correct? And then eventually it leaves by joining, by going through all the complexes and all the coenzyme and cytochrome C and so on. And then it leaves complex 4 by being gained by the oxygen, which is the terminal electron acceptor. However, where does the hydrogen proton go? Now, the hydrogen proton starts accumulating in the matrix. As more taxis come to drop off more electrons, they release more protons, okay? And as these protons continue to accumulate, they accumulate in the matrix of the mitochondria. So in this matrix over here, we're going to find more hydrogen proton. Okay, so every time the NADH is coming to drop off an electron, it's also throwing a proton in the matrix. Eventually, these hydrogen protons are going to build a concentration gradient of very high hydrogen proton concentration in the matrix and very low in the intermembrane space. Okay? Now, as we know, concentration gradients, the main rule for concentration gradients is that the ion will always move from high concentration to lower. So if I start having a buildup of hydrogen protons in the matrix, where are they eventually going to start going? They're going to start moving down their concentration gradient and move across the complex 1, complex 3, and complex 4 into the intermembrane space. And when they reach the intermembrane space, they're actually just going to start accumulating over there. So in the beginning, I had all these taxis coming in, they were dropping off the electrons, and at the same time, they were throwing away the protons into the matrix. The protons accumulated, and when they accumulated, I had a concentration gradient. And eventually, I had a very high H plus ion concentration in the matrix and a very low one outside in the intermembrane space. This caused the hydrogen protons to move across the complex 1, 3, and 4, as you guys can see over here, to go into the intermembrane space. And when they go through the intermembrane space, over time, it's going to be switched. I'm going to start having a buildup of hydrogen protons in the intermembrane space and actually a lower concentration in the mitochondrial matrix. And what's further going to contribute to this is that if you guys remember that when in complex 4, when the terminal electrons, when they reach the exit gate and they want to leave, what takes them in? The oxygen does. The oxygen is the terminal electron acceptor. And so when the oxygen takes up the electrons, it joins with the hydrogen proton and produces water. So it's like the oxygen is further depleting the proton concentration in the matrix. For this is making the concentration even lower than it was before because I have less protons in the matrix, and as a result, I have a much higher concentration in the intermembrane space. And what did I say before? The rule is concentration will always move from high to low. So now it's going to be like, okay, now I want to go back to the matrix because the concentration is just too high for me in the intermembrane space and I can't really handle it. So the H plus ion will move back through a different path, and that will be through the ATP synthase. So this is where the ATP synthase comes into play. It's going to allow the H plus ions to move down the ATP synthase, okay? And this way, they're going to be moving down their concentration gradient. Now, when the hydrogen protons move down their concentration gradient, this releases energy. And this energy is taken up by ATP synthase and used to produce ATP. So this is how we produce ATP. Does that make sense? So we said the whole idea behind this is that, the story is about the chemiosmosis and it's about how we have energy coupling. And what do I mean by energy coupling? I'm going to highlight this in a bit. But just to recap what we've talked about for now, we've discussed that we have 4 parts of the electron transport chain. We have number 1, complex 1, and then we have coenzyme Q, we have complex 3, cytochrome C, and then we have complex 4. The taxis, which are the NADH, they're going to come and they're going to drop off the electron. But to drop off an electron, they need to also kick out the proton. Where do they throw the proton? They throw it in the matrix. And the electron goes to complex 1. The electron will move from complex 1 to coenzyme Q to complex 3, cytochrome C, complex 4, and then it will be taken up by the terminal electron acceptor, which is oxygen. And then oxygen is going to bind with the proton and form water. Okay, so now we've covered the electron part. What about the concentration gradient part for the hydrogen proton? We said that they're being thrown into the matrix. Eventually, they're going to build up a concentration gradient. The H plus ions, the taxis are going to continue coming, dropping off electrons, throwing away protons, until the protons are like, okay, we're way too many in the matrix, we need to make up space. So what are they going to do? They're going to go down their concentration gradient through the through the complexes, complex 1, 3, and 4, and they're going to go into the intermembrane space. Now, when they go to the intermembrane space, over time, what's going to happen as well? Eventually, they're going to also form another concentration gradient where the hydrogen proton ions are much higher in the intermembrane space compared to the matrix. And now they're going to be like, okay, no, never mind. Now we actually, it's like very like, خلاص, we've accumulated way too much. Now I want to go back and I actually want to go into the matrix again. But how will they do that? They can't go back through the complex 1, 3, and 4. They actually need to go back through ATP synthase, and this is where we can produce ATP. So the H plus is going to cross down, and this movement down its concentration gradient through ATP synthase is going to release energy. And this energy is taken up by ATP synthase to produce what? To produce ATP, which was our end goal from the beginning. Okay? So now we've actually covered all these different parts, except an additional thing that I want to add. Okay, I was just talking about NADH, but in the beginning, I said we have two. We have NAD plus and we have FAD. So FAD does not drop off its electrons at complex 1, it drops them off at complex 2. So complex 2 is just an additional, it's just an additional complex. It's not always present. And basically, it just receives electrons from FAD. So FAD drops its electrons where? In complex 2. Just memorize this as a piece of information for you guys to know, just in case they ask you this question. Okay, but it's not a very, like, it's not one of the main complexes. It's just an additional one to accept electrons from FADH2. And this is just some extra information about FADH2, is that FADH2 comes from the reaction catalyzed by succinate dehydrogenase, where when succinate is oxidized into fumarate, it releases the FADH2 because succinate loses electrons and then they're carried by the FAD forming FADH2 and fumarate. So this is basically like this is how we produce FADH2. If you guys were wondering, this is just some like extra thing. Okay. So we have, sorry. So succinate, when it loses electrons, the electrons are gained by FADH2, and then after the loss of the electrons, succinate becomes fumarate. Okay, just this. Very simple and straightforward. And remember that we said that the protons are pumped through the complexes, but complex 2 does not pump any protons because it cannot carry the protons across it. Okay, they're just, so the protons, when they want to go from matrix to intermembrane space, they just go through complex 1, 3, and 4, the main ones, not the additional one. So this is the story I told you guys about, and I explained it here in words, just in case you want to go back to it. It makes more sense here if it's because it's like typed. Yes, and this is me explaining every single thing. So we said the whole story is about energy coupling. What do I mean by energy coupling? It's that the energy from the movement of hydrogen ions down their concentration gradient is coupled to ATP synthesis. And this process where ATP production is linked to the oxidation of NADH and FADH2 is known as oxidative phosphorylation. Why oxidative? Because, why oxidative? Because the electron carriers are undergoing oxidation because they're losing electrons and giving them. Okay, sorry, I think the screen went away again. Okay. So to sum everything up, I said about chemiosmosis is about energy coupling, okay? And what do we mean by the coupling? Important concept for you guys to know in this lecture, and that's mainly that the energy from the movement of hydrogen ions down their concentration gradient is coupled to ATP synthesis, okay? And that this process where ATP production is linked to the oxidation of NADH and FADH2 is also known as oxidative phosphorylation. So why do we call the electron transport chain, and why do we like always mention that, yes, oxidative phosphorylation happens as the electrons move? Is because oxidative, which is when the NADH and FADH2 drop off the electrons of the transport chain because they undergo oxidation when they do, because they're losing electrons and they give them off to the electron transport chain. And what does phosphorylation mean? It means adding a phosphate, okay? And that's how we produce ATP in the final stage, okay? So it's like the first step plus the final stage, and then they form the concept of oxidative phosphorylation. I hope that makes sense. Now here we have a very important force that you guys need to know about. It's that just know its name. We talked about it now and we explained it. But by name, it's called the proton motive force. Proton, which is the H plus ion, and motive force because the concentration gradient created by the hydrogen protons drives ATP synthesis. Because if we did not have hydrogen protons, then they wouldn't go from matrix to intermembrane space, and then they wouldn't have to go back into the matrix and go through the ATP synthase channel to produce ATP, sorry. Makes sense? So it's like they're telling you without the H plus ions, I wouldn't be able to produce any ATP because their movement down is the thing that releases energy for us to produce the ATP in the end. So it's a very vital step. Now here it's just some general calculations for you guys to remember is that one NADH molecule delivers some electrons and this ends up producing 3 ATP, one FADH2, one FADH2 produces 2 ATP molecules. Okay. You guys can remember that NAD comes from vitamin B3, and so it produces 3 ATP. FADH2 comes from B2, so it produces 2 ATP. Okay, so NADH comes from niacin, vitamin B3 produces 3 ATP, FADH2 comes by, and one cycle of transport chain, which we just explained now, creates a problem. Let's have some questions for review. So if anyone would like to answer, which vitamin does NAD plus come from?
B3.
Yes, perfect. And FAD?
B2.
Wow, amazing. Okay. Which complex accepts electrons from NADH? Remember, I told you there's an additional one that accepts from FADH2, and then there's the main one that accepts from NADH. Okay, perfect. So, it's okay. Okay. So these are just, as I told you guys, these are just questions that I've put in between the slides for you just to like test your recall and make sure you're memorizing everything properly. I have actual questions at the end for you to practice the knowledge after like studying everything and understanding it. Now, we're just going to go over all we have left are the electron transport chain inhibitors and the uncouplers, and they're all very easy. Inshallah, don't worry, guys. Khalas, we're about to finish actually. Okay, so now we're moving on to coenzyme Q10. If you guys remember, I mentioned that we have the shuttle buses in between the main complexes. صح? I said there was coenzyme Q. And over here, we just have, yes, you're allowed to answer. Yeah, so we have here, I'm just going to talk about a few clinical, let's say any clinical relations, okay? So we have coenzyme Q10. A very important thing here, the thing I've added in italics is just extra information for you guys just to understand why it does that. But in your lecture, the thing in navy blue was the only thing that was mentioned. So they just told you that coenzyme Q10 supplements may help elevate muscle pain in some people taking statins. Now, when people are taking statins, these statins reduce the amount of coenzyme Q. And coenzyme Q, yeah, this was coenzyme Q. Sorry. Yeah, so this was coenzyme Q, okay? Yeah, I'm just waiting for it to, exactly. So this was coenzyme Q. So as we said, people take so taking statins, when they do take statins, as when a part of the electron transport chain is not there, the electrons aren't able to be transported properly, and hence ATP is not produced properly. So this results in muscle pain because it's like when you're exercising, you need ATP, and when you can't produce this much ATP, your body goes into anaerobic respiration, and when it goes through anaerobic respiration, you'll have lactate and so on, and this will cause muscle pain, and you won't be able to exercise properly. Okay? So this is just the whole idea behind this, is that CoQ10 supplements can help you elevate muscle pain if you're taking statins, because the statins lower the coenzyme Q levels, and this may cause muscle pain because you're unable to produce ATP, which is the main energy currency. Okay? So as I said, the things in italic is just extra for you to understand the idea better. So now we're on to the electron transport chain inhibitors. And drop off the electrons, they would be unable to get. And if I don't have the oxygen taking the electrons at the end from complex 4, then the electrons will remain in the chain to like function properly. And then finally, the last way that they can inhibit the chain is by decreasing ATP synthesis. Yeah, and some of the inhibitors include, number 1, I have rotinone, which inhibits complex 1, cyanide and carbon monoxide, which inhibit complex 4, oligomycin inhibits ATP synthase. So rotinone it inhibits the entry gate, complex 1, the first thing. Cyanide and carbon monoxide can inhibit complex 4, which is the exit gate, the final thing. And oligomycin inhibits ATP synthase. The way they'd ask you about these guys is just by very straightforward questions. They would be recalled. They'll just ask you directly what inhibits this, and so on. Okay. Now to talk a bit about cyanide and carbon monoxide poisoning, the way they inhibit the electron transport chain is that they bind irreversibly to complex 4. And this is very fatal because it's irreversible. If it were reversible, then they just bind and then khalas, you can like decrease the amount of cyanide, carbon monoxide, and hence you can like deal with the problem. But because it's irreversible, this makes it much harder to deal with this thing. And when they bind irreversibly to complex 4, they prevent complex 4 from transferring the electrons to oxygen as a terminal acceptor. And this causes changes in the body similar to when you have hypoxia. And hence, as a result, it's a very deadly situation, and it's not really the best. And just as extra information, carbon monoxide does not only bind irreversibly to complex 4, it can also bind to hemoglobin and display similar changes, such as hypoxia and so on. Because it's like carbon monoxide, wherever it goes, it takes the place of oxygen. That's why it's very fatal. It's going to cause hypoxia and so on, as I mentioned before. Now we move on to the uncouplers. So the inhibitors, they inhibit the chain itself. They act on complex 1, complex 4, and ATP synthase. So they inhibit it. Uncouplers, they mess with the process of oxidative phosphorylation. Because like we said, the electron transport chain has remember when I told you guys the story of how they're coupled, and we have energy coupling, chemiosmosis. It's where I have the hydrogen bonds going down ATP synthase. This releases energy, and this energy is used to produce the ATP, and this is known as the coupling process. So this is what the uncouplers of the electron transport chain work on. So they act on the energy coupling mechanism, and they prevent the oxidative phosphorylation from occurring. And this causes an increased oxidation of NADH, increased oxygen consumption, and decreased ATP synthesis. Okay, so the electrons are still transported. The taxis are still dropping off the electrons because they don't know what's happening at the end of the road. They're still dropping off the electrons at the gate, at complex 1, and they're still moving. And at the same time, the oxygen is accepting the electrons at the end of the electron transport chain. However, it's as if all of this is happening for nothing. As in, there's no ATP being produced, and all the energy that's coming from the electrons moving from one transport to the other is being released as heat. Makes sense? So in uncouplers, it's a very bad situation because the way it happens is that electrons are still being dropped off, electron carriers are still being oxidized, oxygen is being consumed, but no ATP is being produced. And how is the energy lost? It's lost by heat. Okay? Very, very important to remember is that the energy is lost and released as heat when uncouplers are there. So what are some examples on uncouplers? I gave you guys here a diagram just for you to understand how the uncoupling process happens. So normally the H plus enzyme, and we said, would go down the ATP synthase, and then they would produce ATP. In uncoupling, they go down, they can't go down, and as a result, no ATP is produced, and energy is just released as heat. So some examples include, number 1, I have aspirin, and then I have dinitrophenol. which are diet pills, and then I have UCP or thermogenin, which comes from brown adipose tissue. Okay. Now the brown adipose tissue one is important because I have a condition in newborns. So for these three, just memorize them as they are, and just remember what happens when you have uncoupling. Okay. Um, Okay, so for uncoupling protein in newborns, this is an important idea for you guys to remember. And I believe, yani, if they were to get like any type of tricky question or so on, they'd test you guys based on the understanding from these like few slides. So in terms of uncoupling protein in newborns, newborns have a specific thing that we as adults don't have. They have brown adipose tissue in their neck and upper back. This is there for them to, it helps and serves the function of the non-shivering thermogenesis. Because they can't shiver, they need something to regulate their temperature, and as a result, they have the brown adipose tissue. Because for us, when we feel cold, we just shiver. For them, they have the brown adipose tissue to help them do that. However, in brown adipose tissue, I have uncoupling protein. It's also known as thermogenin, okay? So this uncoupling protein, from its name, it causes uncoupling, okay? But it's only activated in cold temperatures. So when I have cold when the baby and senses cold, their UCP1 is activated, which is the uncoupling protein. And when it's activated, it causes uncoupling of the oxidative phosphorylation, and as a result, the protons will start going down the ATP synthase channel. However, they're going to start going down the uncoupling protein. As a result, they will not be producing any ATP. They're just going to be releasing the energy as heat. Okay? So remember this. New uncoupling protein in newborns, where is it found? It's found in the brown adipose tissue that they have in order to control their non-shivering thermogenesis. This uncoupling protein is found in the membrane, in the inner mitochondrial membrane. And when a baby is cold, this UCP1 is activated and it causes uncoupling. As a result, they stop producing ATP and the energy is released as heat. So now it's time for questions. Whoever would like to answer, go ahead in the chat or just open the mic. And yeah, let's begin with the questions. So, biological oxidation reduction reactions always involve which of the following? Direct participation of oxygen, formation of water, mitochondria, transfer of electrons, transfer of hydrogens. Which one is it? Correct. Now, moving on to question 2. Which of the following components of the electron transport chain is inhibited by oligomycin? Yes, perfect. ATP synthase. Okay. Which of the following best explains how the brown adipose tissue in newborns generates large amounts of heat? Does it cause an increased availability of fat, or increased formation of ATP, or activation of UCP1 in the mitochondria? Remember the UCP1, the uncoupling protein. What do you guys think? Perfect. Go ahead to question 4. So you're carrying out experiments and you add the electron transport inhibitor, cyanide. You then examine the oxidation state of various components of the electron transport chain. Which of the following would be expected to be in an oxidized state following this experiment? Now, there's a few stages to this question. I'm going to give you guys time to answer, and then we can explain it. Are you saying C for this question, or you meant the one before? Oh, okay. Do you want me to explain the question first, or you know the answer? It's okay, I can explain. Okay, so this question has a few, let's say, layers to it. And we'd first start, let's understand the situation. So they're telling you you're doing an experiment and you added an electron transport inhibitor. What do the inhibitors do? They act on a specific component and they just inhibit it. So cyanide, if we remember, inhibited which part? Complex 4, exactly. So cyanide, just as well as carbon monoxide, they inhibit complex 4. Now they tell you that you then examine the oxidation state of various components of the electron transport chain. Which of the following would be expected to be in an oxidized state? Now oxidation is what? Is oxidation loss or gain of electrons? Exactly. So oxidation is loss of electrons. So for how many components we have? We have 1, we have coenzyme Q, and then we have 3, and then we have cytochrome C, and then we have 4, okay? So they're asking us which one is going to be an oxidized state. So for it to be an oxidized state, it could either have transferred an electron to the next component or never received an electron, صح? Because oxidation, you just don't have electrons. But let's go back to the complex. Okay. Where are the complexes? Okay, here. So they're telling you which one is going to have lost electrons. Yeah, exactly. So we have it. Actually, just a second. I think I answered this the wrong way. Okay, let's skip over this question. I should have added a piece of information in it, honestly. عشان it's missing something. Okay, let's do question 5 and then I'll fix the one and then I'll attach it in something else. Okay, so for question 5, it says the electron transport chain in the mitochondria contains ATP synthase that convert the energy of oxidation reduction reactions into a form that can be used for the synthesis of ATP and for the uptake of solids. This form of energy is most likely a result of the following: proton gradient, sodium, and potassium gradient. Okay, which one is it? This is an easy question, relatively. Yes, it's A. Perfect. But for question 6, what's the answer? So someone said D. Yes, that's correct. MashaAllah, you guys are getting them all correct. So question 7 says, according to the chemoosmotic theory of how oxidation and phosphorylation occur, are coupled, sorry, which of the following processes produce the chemical gradient that drives the generation of ATP from ADP? Would you guys like me to explain this question? Okay. So in question 7, they're telling you, explain, okay, how oxidation from coupled. Exactly. So they're asking us basically here about the chemical gradient that produces ATP. Okay, what do they mean by that? They're saying that, number one, first option says electron transport causes H plus to penetrate the inner mitochondrial membrane. Does this apply? Is this correct? No, because H plus does not penetrate the inner mitochondrial membrane. It goes down its concentration gradient through the complexes. So A isn't correct. And then B says, electron transport ejects H plus into the intermembrane space. And this could be a possible, it could be a possible answer, okay? And then we have FADH2 is regenerated from FAD. This has nothing to do with the chemical gradient. And then we have NADH is regenerated from NAD+. This also has nothing to do with the chemical gradient. And then we have osmotic forces cause H+ to bind with FAD in the mitochondrial matrix. And this is completely incorrect. This is irrelevant. So we end up with B. Now, why is B correct? I will explain to you guys. So basically, the normal flow of electrons through the proteins of the oxidative phosphorylation is coupled with the proton gradient across the inner mitochondrial membrane. So as the electrons move from one complex to the other, they release this energy, and this pumps the protons out of the matrix into the inner membrane space, okay? And then we create here a proton gradient, and this proton gradient is what drives the production of ATP. So makes sense? So if the electron transport wouldn't eject the H+, I wouldn't have a gradient, and as a result, the H+ won't move through ATP synthase, and hence I would not produce ATP. So this is just a complex way of asking you guys which gradient is responsible for producing ATP. Make sense? Okay. Now I go back to the question above, the one that I just skipped over. It was question 4 بالضبط. Now for question 4, we want to think of the complexes. طيب, so let's go back to it. We said that we're carrying out experiments, and then I add an electron transport inhibitor, which is cyanide. Cyanide, we said, inhibits complex 4. And they're asking you, if cyanide is added, which of the complexes will be in the oxidized state? طيب, oxidized state meaning that it does not have any electrons. So let's say lost electrons or no electrons. Now, in your opinion, which one do you think, if you guys know? If not, عادي, I'll just continue. But if you do know, just put it on the chat so I can that you guys are understanding the points I'm making. Yeah. So in this answer, I believe that part A would be the correct one, okay? Why is that? Because let's think of it. Complex 1 has no problem. It's gonna give off its electron to coenzyme Q. But then coenzyme Q is gonna give it to C, and then 3 is gonna give it to cytochrome C, صح? But when it goes to from cytochrome C to the last complex, which is complex 4 is gonna be inhibited. And as a result, it will not receive any electrons. Hence, this goes back to the fact that complex 3 will not, cytochrome C will not have lost any electrons, and as a result, complex 3 as well will not be in the oxidized state, and this leaves coenzyme Q in the oxidized state. I will reword the question just to make sure that it's clearer and more straightforward, and I'll add the explanation when I share the presentation. So that's all for today. I hope you guys benefited, and I hope you really understood the electron transport chain. And you can scan the QR code for feedback and let me know what you think of this session. I hope it was well.